Alright, hello everyone. The goal of this video is to learn how to complete a dihybrid genetics cross. I'm going to walk you through the steps, and then you can refer back to this when completing problems on your own. So first things first, let's define what it is. So basically, after Mendel did his work with the monohybrid crosses that we looked at before, he wanted to know, okay, well now instead of looking at one gene, what happens if I track the inheritance of two genes? What sorts of patterns can I observe? So a dihybrid cross is a cross that tracks the inheritance of two, hence the dihybrid genes. So he wants to know over generations what happens to the two genes. So he really ran the same experiment as before, P generation, F1, F2, but now looking at two genes, and that's what we're going to do. So here we have our key. So let's just draw out the alleles and just say what they mean. Uppercase Y stands for yellow colored seeds, and lowercase y, I try to draw these a little different looking from each other so I don't get confused later, stands for green. And then the seeds can also have different shapes, so that's the other gene. Now we have the color gene here, so let's just put a little note, that's the color gene. And the other gene we're going to look at comes in two versions, or alleles. They can be round, or they can be wrinkled. And that's the shape gene. So you have two different genes you're looking at, color and shape. So that's why it's a dihybrid cross. So now the genotypes are similar, but they're going to look a little different because we're putting two genes down. So here it says it wants us to cross a homozygous yellow round seed with a homozygous green wrinkled seed, and then it wants the genotypic and phenotypic ratios. Alright, so same steps as before when we did a monohybrid. First things first, figure out the male genotype. So the first one, the male, I don't know if they say. So the male we'll say is homozygous yellow round. So that would be homozygous yellow, that's the color gene, and homozygous round, the two dominants. So that's the first genotype. Two genes, 1, 2. Next one, the female, is homozygous green wrinkled. So lowercase r's, lowercase y's. Now, to create the gametes here, the sex cells, we have to write all possible combinations. So remember, law of segregation, I take one allele from each gene. So I'm going to take this uppercase Y and this uppercase R, both put them there. Now this is not a genotype, it's just showing me which alleles the sex cell is carrying. I could do more of these, but I don't really need to because there's only one possible combo from each gene, an uppercase Y or R. Same here, except lowercase. And now I have the sex cells, I can see what they are carrying. So do I need a Punnett square for this problem? I could set one up, but I don't really need to because I kind of know what's already going to happen. If we look at each gene separately, if I take this sperm to this egg, I'm always going to have big Y with a little y, and uppercase R or big R with lowercase r every time, no matter what. So there's no need to do a whole square. You could if you wanted to check yourself, but you're going to start repeating a lot. So the offspring are pretty much going to be 100% heterozygous, just like we saw in the monohybrid. So still law of dominance, yellow is dominant over green and round is dominant over wrinkled. Phenotype, all the seeds from these two plants are going to be yellow and they're going to be round. 100%. So not too bad. So now we got our F1s. We're going to do the same thing over. We're going to cross this with itself or with the same genotype and see what we get. And that's where this side comes in. And it gets a little trickier here, so pay attention. So if two offspring from the previous were crossed, what's going to happen? What are our genotype and phenotype ratios? We'll focus more on the phenotypes. No need to go crazy with the genotypes. So first things first, let's write out our parent genotypes. You always want to write these out just so you know what you're crossing. So now, same from the last one, right? We're taking the F1, crossing the same ones. Now, this is one of the tricky parts. When we do segregation, there's another thing going on here. The way that the alleles go into the sex cells is random, and they do this independently. So that means that the big Y and the big R don't always have to be together, and the big Y and the little r can go together, or something like that when they enter the sex cells, because the chromosomes all segregate randomly. So we need to do every possible combo. So one way you can do that is just by looking and trying to get every combo. So I'm going to take this Y and this R. Remember, two different genes here, so I take one from each. Take the Y and I take the R. Uppercase. Then I'm going to go to a next possible combo. Uppercase Y, but I'm going to use a lowercase r. Now the other way around, I'm going to use an uppercase y, or excuse me, a lowercase y and an uppercase r. So remember, this is not a genotype, so I can put the little y first. I'm just showing what alleles are being carried by the sex cells. And there's my last possible combo, the two little ones. Now, if that doesn't work great for you, you can use a method that's probably similar to something you've heard in math class called FOIL. And some people like to use this. It's one way to kind of prevent yourself from missing anything. We'll take the first. So remember, these are the two genes. So I'll take the first letter from each gene. So big Y, big R. The outer, so Y, big Y, little r. The inner, so little y, big R. And the last, little y, little r. So that's all my possible combos. So same thing, I can use FOIL to help guide myself. But you really just want every possible combo of the big and the small from each gene that you can get. Now we have to set up the square. Same as before, except a little different. You're going to put two letters over each box instead of one, because remember, it's a dihybrid cross, so there's two. So two alleles. So big Y, big R, big Y, little r, lowercase y, uppercase r, and lowercase, lowercase. Same exact thing on this side. Really doesn't matter which side you use, especially in this case, because they're all the same anyway. Actually, it would never matter. So that same thing. I'm just going to keep the same pattern I used on the top, the two recessives going last. So now I have the sex cell set up. The last thing I need to do is recombine these through fertilization. So let me go ahead, and it's going to take a minute. We'll just go through all these. So big Y, they're going to meet here when this sperm and egg meet. So now I'm putting the two letters back together, because that's what the sperm and egg do, right? They make the organism diploid again. They add the two halves back together. So same here. There's a possible combo, uppercase Y, uppercase, that. This one down here, this one over, this one down, this one down, this one down there, and I'm just following this, and I'm just going to keep going now and finish the square up. Remember, I'm just dragging these over in my mind. I'm just saying, okay, that big Y there, this little y here, that big R over here. Always put the uppercase first for each gene. It's the fun part. Alright, so there's all my possible genotypes for the F2 generation. Now I'm just going to go for my phenotype ratio. So I kind of already answered this. Here's the number of different genotypes. They're all there if you want to count them. There's 16 boxes. If you ever have to draw one of these squares, it's a box with 3 lines down, 3 lines across. So 16 squares total. So let's do our phenotypes. There's 4 different options I can get. I can have the 2 dominants in one seed, so yellow, round. It's one option. I could also have a dominant yellow with a recessive wrinkled. I can have recessive green with dominant round, and then the last possible combo is the 2 recessive, so green, wrinkled looking seeds. So now I just have to use the square. I'll start with these, and I'm just going to draw a little circle in each one that I find. I'm looking for anything that has uppercase Y's and uppercase R's. So this one would work. This one would not work, right? There's a lowercase R's. So that one, that one, that one. Not that one, right? This one, this one, this one, and no. So everyone with a circle counts, so I'll just count them up. 1, 2, 3, 4, 5, 6, 7, 8, 9 out of 16 boxes. So I'm just going to put 9 and start building my ratio. Next, I'm looking for anything with uppercase Y's and lowercase R's. So I'm going to put a little line in that one. So here's uppercase Y, lowercase R's, uppercase Y, lowercase R's. No. Here, this works. And no, these are all lowercase Y's. So 3. Next, anything with lowercase Y's and uppercase R's. So this one works, this one works, and this one works. So I got 3, and then out of the whole 16 I get 1 out of 16 for the recessive. So my ratio is 9, 3, 3, 1. So did the big Y's and the big R's have to stick together? No, right? We can show that with the ratio. The 9, 3, 3, 1 proves one more law of Mendel's. He called this the law of independent, hopefully you guys can see this, assortment. And the law of independent assortment just explains that the alleles are able to independently assort, right, or move into the sperm or egg cells. And again, Mendel was describing meiosis. He didn't really know that, but that's essentially what he was doing. So use this. You can pause it, do what you need to do to help yourself answer the dihybrid problems. Common things that people make mistakes on is, remember, you use the 2 different alleles from the genotype, and they recombine together, keeping those same letters next to each other when you do the genotype. So just follow this, make sure you're careful about that, and you should be good. If you're not sure, you can always go back. One thing that's nice is whenever you see the 2 parents are heterozygous, big, little, you always know you're going to get the 9, 3, 3, 1 phenotype ratio. You can always do a square to test yourself. So please email, and if you have any questions, I can always go over a problem with you, or Miss Paulson can if you're in her class. So good luck, and thanks for watching. Bye.